Fine. Okay, we will solve this exam. I chose a random exam. Now tell me, let's be quick. The main component of milk is a liquid that is important, an important solvent in the body. What is the name of this solvent? Any solvent in the body, what is it? What does everything dissolve in?
Water.
Water, right? Let me see. Okay. Let me go back to the exam. Yes, correct. I'll be quick so that we finish and I'll let you go. Uh-huh. Yes, water. Okay. What are the elements present in protein? Quickly, be quick. I want to finish this exam quickly. What are the elements found in protein? Any one of you give me the answer. Protein is made up of what?
Nitrogen, carbon, hydrogen.
Yes. Yes, they are all made up of hydrogen and carbon and oxygen, except protein. We have the additional nitrogen and sometimes sulfur. What is the enzyme that breaks down protein? Khadija, give me a name of an enzyme that breaks down protein and the products of digestion. This is so easy.
Protease.
Protease enzyme, correct. Like pepsin, like trypsin. And what are the products of digestion?
Amino acids.
Amino acids. Very good. Now the next one. Fats. Give me an enzyme that breaks down fats quickly.
Lipase.
Lipase.
Lipase. And fats are made up of what?
Fatty acids and glycerol.
Fatty acids and glycerol.
Fatty acid and glycerol. And the elements that make up fats?
Carbon, hydrogen, oxygen.
Yes, let me just open this from here. Okay, very good. Okay, lactose. Now, lactose, this is supplement. What are the elements that make up lactose? It's lipase. Give me an answer quickly. Fatty acids and glycerol, and the elements in fat, carbon, hydrogen, and oxygen. No nitrogen. Now, the lactose, it's made up of carbon, hydrogen, and oxygen also. What is the name of the enzyme that break down lactose? We took it in biotechnology.
Lactase.
Very good,
Lactase.
lactase. Lactase, excellent. Okay, I'll highlight this so you don't forget it. Now, explain why vitamin D is important for the nutrition of children. This is year 9 material. Why is vitamin D important?
For strong teeth and bones and prevent rickets.
Yes, for strong teeth and bones. Very good. Very good. This is enough. Also, let's just give more information because we are revising together. What does vitamin D do? It helps in what? So that we have strong teeth and bones. It helps in the up. Hmm. What is the information that you took about vitamin D? We took more information than what you said. We said that it helps in the absorption of.
Calcium.
Very good. Let's remember it together. So it helps in the absorption of calcium in the body, and it is activated by.
Sunlight.
Yes, it is activated by sunlight. And of course, because calcium is absorbed in the body, it is important for the strength or the health, just like you said, bones, teeth, and without it, you will have rickets in children or weak bones or osteomalacia in adults. Very good. Explain why iron is important in the diet. Again, this is year 9. Why is iron important in the diet? Very easy.
Responsible for the making of the protein hemoglobin that transports
I can't hear you.
oxygen.
Red blood
Very
cells
good.
produce more hemoglobin.
Excellent. Hemoglobin, more red blood cells, and hemoglobin and red blood cells are needed for what?
Transport of
To
oxygen.
transport Very good. Transport oxygen. And without hemoglobin and red blood cells, what disease will you have?
Anemia.
Anemia. Okay. Now, I don't want you to write just 2 points because they are 2 marks. I know that you all do that, but write the rest. Why not? Because I'll tell you why. Yes, they are 2 marks and they want 2 points, but which 2 points you don't know. Maybe you will write that the iron is needed to make hemoglobin, to make red blood cells, and you stopped. What if they consider that these two are 1 point? You know the rest of the information, so write it down. It's not going to take like a second. So you look at the marks and you tell me they are 2 marks. I'll write 2 points. Now, why don't you write three? Why don't you write four? The more, the better. Why? Because they can count two as one. If you tell them that I need the iron because it will help me produce hemoglobin and red blood cells, they are two separate points. You considered them two separate points. What if they consider both as 1 point? So you take one out of two. It's such a waste of marks. You already know the rest of the information. So when you know the rest of the information, write it down, even if it is out of 2 marks. Okay? Because they can count hemoglobin and red blood cell as 1 point, or they can count it as two. We don't know. Good? Let's look at the mark scheme. I want to tell you, I want to show you how they grade you because grading changes from one year to the next. The same question can be graded differently in different years. This is question 1B. Let's go to 1B. Okay, 1B double I. Look, it is required to make hemoglobin. Let me activate the pen. It is required to make hemoglobin / red blood cells. So this is 1 point. Just like I told you, this is 1 point. And then give another point, transports oxygen. This is another point. Prevents anemia. This is a third point. So if you tell me I wrote 2 points, I said it is needed in the making of red blood cells and in the making of hemoglobin. Excuse me, yes?
I can't see the share screen.
Oh, 1 minute. You cannot see the shared screen. Why is that? Can you see it now?
Yes.
Okay, I'm showing you the mark scheme and I'm teaching you not to write exactly the same number of points, but write more. Now, exactly like I told you, here, the iron, it is needed to make hemoglobin and red blood cells. These two are 1 point. So if you told me, doctor, I wrote 2 points, why did they take one out of two? Because when you wrote it is needed to make hemoglobin and it is needed to make red blood cells, they counted the two as 1 point, one out of two. So to be on the safe side, write the rest of the information. Okay? I'm just giving you a tip and it is your decision. Good. Now, define the term transmissible disease. This is also year 9. So year 9 is important. Please do not ignore it. What is a transmissible disease?
It's a disease that can pass from one host to another.
Yes, this is 1 point. It is caused by what?
A pathogen that moves from one host to another.
Yes. You have to mention that it is caused by a pathogen because we have other types of diseases, like genetic disease. A genetic disease can pass from one person to another. Something is genetic. Yes or no?
Yes.
Genetic, like autism, cancer. Is it caused by a
No.
pathogen? No, it's not caused by a pathogen, and it is not transferred from one person to another. So a transmissible disease, why are they out of 2 marks? Because you have to mention that it is caused by a bacteria, a virus. It is caused by a pathogen. And because it is caused by a pathogen, this pathogen can move from one person to another. But if you have, God forbid, cancer, this cancer cannot go from one person to another. It is not transmissible. Pay attention to that. Explain why women should be vaccinated against rubella before they reach reproductive age. Now, some of you took a picture of this and they told me we don't know what rubella is. You don't have to know what rubella is. Why should you be vaccinated? Why should girls be vaccinated before they reach reproductive age? And by the way,
So
they
what
did already give you information that rubella is a transmissible disease, and ignore kwashiorkor, it is a non-transmissible disease. And rubella is a disease that affects children. So why should you be infected with rubella when you are pregnant and you are vaccinated? Okay, I don't know why we lost connection, but tell me, give me, this is an important question. Why should a woman be vaccinated against a transmissible disease before they reach reproductive age? 2 marks, 4 marks.
to prevent passing rubella to children?
Yes, number one, they said that rubella is a transmissible disease. So it is caused by a pathogen. So it can be transmitted. Okay, so number one, you will mention that we don't want the rubella to pass to the baby. How does it cross to the baby?
Placenta?
How- Very good. So the rubella is a transmissible disease. So it can reach the baby. Number two, rubella, by the way, is caused by a virus and it is also in year 9 notes. Number two, it can cross the placenta. So we don't want this to happen. And if it crosses the placenta, is it healthy or harmful to the baby?
Harmful.
Of course, it is very harmful to the baby. Okay, inside the womb, it is called fetus. Number four, why do we get vaccinated? How will vaccination help you? Talk a
مش
little
مش
bit about
rubella ده
vaccination.
مش rubella ده because retardation.
Yes, if you have or you know any information about it, you can also write it is caused by a virus. It can cause retardation to children. It's very harmful. It crosses the placenta. The placenta does not provide 100% protection. So we will become vaccinated. Now, why will we be vaccinated? What will the vaccine give you?
It will produce
Will
memory
create memory
cells.
cells.
Exactly. You are all correct. Because you know very well that the vaccine is the weak virus. It will activate the immune system. Lymphocytes will produce antibodies. The antibodies, some of them will remain in the blood and give you memory cells. These memory cells will give you long immunity or long lasting immunity. So you have now memory cells in your blood. If the mother, while she is pregnant, she is infected with the rubella virus, what will her body do? We have memory cells in our body, and you are infected with the rubella virus. These memory cells, what will they do?
They will directly kill أو mark الباثوجين.
Very good. Yes, they will become activated very quickly, divide by mitosis, produce plasma cells, and these plasma cells will produce antibodies. And the antibodies, they will work against the rubella virus. Just like you said, they can mark it, they can directly kill it, whatever, so that you are immune. You will never become sick. And if you are not sick, you will not give this virus to your child, and you will be safe, and the child will be safe. So this question is out of 4 marks. Now you need to focus on questions that are 3 marks and above. Here you can write a lot of things. Here we are talking about a pregnant woman and we want to protect her baby. And we are talking about vaccination. And these are two topics that you studied very well. So you can discuss this and that. You can at least mention 2 points about the protection of the baby and 2 points about vaccination. This is how you will distribute your information. You see 4 marks. And here you will underline while you are reading the question, the word vaccination, and you will underline the word rubella. And they told you that rubella is transmissible. And they told you that here the mother is protected, or we are trying to protect a baby. So the first thing that should cross your mind is protecting the baby by the placenta. You have to write this down. And that rubella is a transmissible disease caused by a pathogen. It can reach the baby. And you have a lot of things to talk about vaccination. Okay. The problem with the essay paper is that you know the information, but you don't write all the information down because the questions are not direct. The questions are misleading.
Doctor, I can't see the share screen.
Oh, I don't know why we are losing connection. I was speaking to myself. Okay, the causes of kwashiorkor is cancelled.
This the screen isn't shared.
This is so annoying. I was saying something really important. I don't know if you heard it or not. Okay, let's move on. Now, question 2. Obviously, it is here showing you a particular process. Which process here? Here I have a DNA, and then this DNA will, there's an arrow. What is this? What is A?
Messenger mRNA,
Yes, this is the messenger RNA. And what is structure B? What is B?
ribosome,
Yes, it's a ribosome. If anyone disagrees with me, tell me. This is which process? Messenger RNA, and it will go to a ribosome. This is the process of what? Who will remind me? Yes, this is protein synthesis. Okay, explain the role of the molecule A in protein synthesis. 3 marks. We said that A is the messenger RNA. So what does the messenger RNA do? What is its function?
it copies parts of the gene and it determines the type of protein.
Excellent. It will do the transcription part, or the copying part of the DNA that we want to produce. And then does the DNA stay in the nucleus or it goes out of the nucleus?
It stays, it's only copied.
It stays. Very good. It is copied by the messenger RNA. The messenger RNA will leave the nucleus and it will bind to a ribosome. And the ribosome is going to read the sequence of bases on the messenger RNA. And don't forget this very important sentence, that the sequence of bases on the messenger RNA, it will determine the final sequence of, the final order of
Amino acids,
amino acids. This is a very important sentence. Always mention it when you talk about protein synthesis. What is the role of insulin? Why is insulin important? 4 marks.
decrease
4 marks.
the sugar glucose blood
Maintains blood
Yes.
concentration.
glucose levels.
Yes, I want you all to answer. Very good. It will control sugar levels, maintain sugar levels, correct. Now, it will increase it or decrease it?
Decreased,
Very good. It will decrease sugar levels. This is the first point you need to mention. It's a hormone that decreases sugar levels. How does it do that?
it stimulates the liver,
Very good. It activates enzymes in the liver to do what?
to convert glucose into starch, into carbohydrates.
Not starch. Not starch. Do we store carbohydrates as starch as humans?
Glycogen.
Fats,
Very good. Okay, now the insulin does a lot of things actually. The insulin, it lowers the glucose levels. This is its main function, and it does that by three ways. Number one, it will go to the liver, activate enzymes in the liver, and tell the liver, please take the excess glucose and store it as glycogen, just like you said, the glycogen. Now, the liver stores have been finished. I don't have any more space in the liver. What else does it do? We took all the 3 points.
It will convert it to fats under the skin.
Very good. It also helps in the conversion of glucose to fats, and this is stored under the skin. That's why they tell you while you're dieting, you have to keep your insulin levels low because insulin makes you fat. And also it increases the rate of absorption of glucose by the body cells in general, so that they use the glucose in respiration. So it increases the rate of respiration. You can write all three if you want. The most important is the conversion of the glucose into glycogen and storing that in the liver. Now, this is 4 marks. What else can you write about hormonal regulation? This is an example of what?
Negative
Positive,
feedback.
negative feedback.
Yes. Anytime you are discussing hormones and hormones are regulating something, always write it as an example at the end of the question. And this is an example of homeostasis by negative feedback. Okay. Now, in case they are asking you another question, in case they are asking you about homeostasis by negative feedback, a tricky one, a tricky question. If the insulin levels are high, which hormone will be low?
Glucagon.
Yes. Okay, because hormones, they work in pairs. It's like they are antagonistic. One is high, one is low. One is low, one is high, because their actions are the opposite. That is why one is high and one is low. And they are controlled by this manner. When the brain detects that one of the hormones is high, then the other should be low, and vice versa. And this, or any hormone function, always at the end, after you speak about any hormone, the last sentence, this is an example of homeostasis by negative feedback. If you discuss any hormone, okay? State the names of two substances other than insulin that are secreted by the pancreas.
Glucagon, amylase.
Very good. Glucagon is the opposite hormone. Okay, we just mentioned it. It is released when the glucose levels are low and we want to increase them. And any enzyme, because the pancreas releases pancreatic juices that contain a collection of enzymes: lipase, pancreatic amylase, maltase, trypsin. Don't forget, trypsin is produced by the pancreas and the epithelial lining of the small intestine, while pepsin is produced in the
Stomach.
correct. Now I have here a diagram. Of course, now we are, so that we don't waste time, we go directly to the question. But on the exam, you read the whole question from beginning to the end. Explain why temperature has an effect on the rate of photosynthesis in general. Why does temperature has an effect on photosynthesis? 4 marks.
It increases the rate of the reaction by more successful collisions on substrate, enzyme substrate complex. كل ده. Kinetic
Yes,
energy.
كل ده. Yes, that is the most important thing. Also, they can ask you this question in a different way. Why does temperature affect respiration? Why does temperature affect photosynthesis? Why does temperature affect any reaction in the body? Because any reaction in the body is controlled by what? What's the first thing that you should say before saying all that? Yes, any chemical reaction in the body is controlled by enzymes. Any reaction. We have lots of reactions in our body, but we did not study except respiration. So any reaction, you have to mention or start the answer by mentioning enzymes. Any reaction is controlled by enzymes, and enzymes are very sensitive to temperature. What happens to enzymes at low temperature and at optimum temperature and at high temperature? You will give a very good answer. At low temperature, what happens to the enzymes?
Work slowly.
Yes, they slow down. So the reaction will be very slow. And this is something that we don't want. As the temperature is increasing, just like you said, it will increase kinetic energy. We said it 100 times. Anytime you are discussing temperature and enzymes, you have to mention kinetic energy. So as the temperature is increasing, reaching optimum, kinetic energy is increasing. We have successful collisions, like just Selma said, very important keywords. There is more and more formation of enzyme substrate complex, faster reaction. We will reach optimum temperature, maximum rate of reaction. We know this very, very well. What if the temperature is very high?
Enzymes denature.
Enzymes will denature and the reaction will completely stop. Okay, we are talking about photosynthesis. So when the temperature is optimum, there will be maximum photosynthesis. Maximum photosynthesis. This is very good for the plant because the plant will produce food and it will grow and etc. 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 Good. And also photosynthesis here involves the exchange of gases. And gases, how are they exchanged? How do they move into and out of the plant?
In diffusion.
By diffusion. And the higher the temperature, the faster the diffusion. Okay. So this is a very direct question. It's very easy. And this question they can ask you about photosynthesis, respiration. The answer is nearly the same. Explain why the rate of photosynthesis will decrease if the humidity and the apparatus becomes very low. Now, this is a little bit of a tricky question. The humidity becomes very, very low. So
No
why will the
gradient.
rate,
No gradient.
excuse me, wait, Yusuf and Omar, can you please give me an answer to this question? We have very little humidity. Why will this make the rate of photosynthesis very low?
It will decrease the water concentration around the leaf, so the stomata will
Very good.
close.
Excellent. You have a leaf, and outside the leaf it is very dry. We have low humidity. So this means that the water potential inside the leaf will be very high, and the water potential outside the leaf will be very low, just like Yusuf said. And the water gradient or the water concentration will be very different. Very high inside the leaf and very low outside the leaf. And this will increase what? When I have very little humidity outside, it's very dry outside. This will increase the rate of,
Transpiration.
correct, water vapor will go out. This will increase the rate of transpiration. Now, too much transpiration, water out, water out, water out, the plant will wilt. In order to prevent wilting of the plant, the stomata will, someone said it,
Close.
Close.
Yusuf said, yes, the stomata will close. Stomata will close. There will be a decrease in the exchange of gases, and a decrease in the exchange of gases means a decrease in the movement of the CO2 inside the plant and less photosynthesis. So this is a tricky question. You know the information, but again, they did not ask you a direct question. They did not ask you, why will the rate of photosynthesis be affected when the stomata are closed? They did not ask you a direct question. This is the problem with the questions, that they can sometimes be tricky. So I'll highlight this for you, but you did a good job and you answered it very well. Okay. Now, explain how scientists would use the readings for the concentration of carbon dioxide in chambers 1 and 3 to calculate the rate of photosynthesis. Now, calculating the rate of anything. How do we calculate the rate of anything?
هنعمل ال difference between chamber 1 and chamber 3 و 1 over time.
Very good. The rate is the difference over the time. Perfect. Now, graphs and graphs and more graphs. I have temperature on the x-axis and the rate of carbon dioxide uptake and release. So you have two graphs. You have a key. Here you have the crosses is the uptake of carbon dioxide during the light. And the dashed line is the release of carbon dioxide in the dark. Okay, the rate of uptake of carbon dioxide in the light in 20 degrees. So this is so easy. I'll go to 20 and I'm going to look at the rate of uptake of carbon dioxide. Which graph is the uptake? This one. So you will go to 20 and you will move up until the graph stops you, and then you will give a reading. What is the reading here? Now they want to see if you can read a graph. Tell me, this is 120 and this is 140. So there is a value of 20 between the two boxes. And how many boxes do I have? 5. So every box has a value of how much?
4.
Yes, 4. So when it is 1, 2, 3, what's this value? This is 1, 2, 3, and 4. 4. So 120. This is what's the value here? 4 times 4. This is 16. 120 plus 16. What's the answer? 120 plus
136.
16? Yes, 136. They just want to see if you can read a graph. Now, the rate of release at 20 degrees, the rate of release is here. This is very easy. It is directly 20. Okay, so you should be good at reading graphs. Explain why the rate at which carbon dioxide is used in photosynthesis is greater than the rate of the carbon dioxide uptake. You guess? I am absorbing carbon dioxide, but I am using more than what I am absorbing. Mm. Where did the rest of the carbon dioxide come from?
And
What will you
stored.
say here? Yes, I have carbon dioxide already in the body of the plant because the plant is always respiring morning and night. Okay, so the carbon dioxide is already in the body of the plant. The body of the plant already has some carbon dioxide because the carbon dioxide is produced by what? By, give me an
Inspiration.
answer. It is reproduced by respiration day and night, all the time. So even if I do not absorb any carbon dioxide, I already have some carbon dioxide in my body, and I'm going to use it in photosynthesis. Explain why the results in the table are expressed per centimeter squared of leaf rather than per leaf. By the way, these are tricky questions. They depend on critical thinking. Anything depends on critical thinking, I get very funny answers. Now, why would they measure the area of the leaf as per centimeter squared rather than per leaf?
علشان it's one leaf.
No. One plant has one leaf. Of course, one plant has many leaves. Are all the leaves the same size?
No.
No, all the leaves are different. I have a big leaf with large surface area, small leaf with small surface area, because the leaves are not the same size. Not same size. They're not the same surface area. Okay. Of course, it's more accurate this way. I can compare between the surface areas of the leaf and the rate of photosynthesis in both. This question also is tricky. Okay. Ecology. It discussed the advantages of growing crops in glass houses. You did not study glass houses. I know glass houses, but they gave you information. You expect something like this in any subject. They always give you something new and they want your opinion about it. So they told you here that some crops are grown in controlled environments. Glass houses are an example of a controlled environment. Figure 3.3 shows a glass house where lettuce are grown. So what is a controlled environment? They will not get you a question about glass houses, just be sure, except if they give you information about it. It's a house, and inside this house, everything is controlled. I'm controlling the temperature, the pH, the water, the fertilizers, the light, the carbon dioxide concentration. Everything is controlled. So what are the advantages? What will you say? Out of 6 marks.
Plants not affected by pesticides or insecticides yet.
Yes, plants are not affected by anything. Outside in the garden, the plants can be exposed to insects, to pests, which are the same thing, to very harsh environments, to little rain, to drought, to anything. But inside a glass house, it is a perfect environment, like a perfect world. Remember the fermenter? When we used to grow bacteria in a fermenter and we said that we will make the bacteria happy, how did we make it happy? Hmm?
Provided with suitable temperature, water.
Exactly. We will give the bacteria everything it needs so that it will reproduce. And the same idea as a glass house. I'll give the plants inside the glass house everything it needs so that it will do maximum photosynthesis. And there will be no anything that will prevent photosynthesis. What is it called?
Limiting factor.
Exactly. This is so important. Now, this is out of 6 marks. So if I am new, I am going to discuss 6 points that will be provided to the plant to make the plant happy. So this controlled environment or glass house, number one, it will have no limiting factors. The plant will be happy. I'll give it light, exactly the same or the best concentration of light. I'll give it proper temperature. I'll give it fertilizers. And I will discuss the mineral ions here. And I'm going to Okay, Yusuf and Khadija, can you tell me what is a limiting factor?
A factor in the reaction,
Mm.
if it's all used up, the reaction will stop. It's
Okay,
the
it's a
factor
fact.
that is
Mm. Okay,
it's the
what you
factor that
said
will
is
stop the
correct.
reaction.
Yes, what you said is correct. Is the factor that if it is not present in proper amounts, it is going to affect the photosynthesis, the reaction of photosynthesis in the glass house. Do we have limiting factors or no?
No,
Yes or no? Exactly. I'm going to give the plant maximum light exposure. This light is important for what? And how can I give it? Think this light is important for maximum photosynthesis, and I can adjust the light intensity here by using light bulbs, for example. Temperature. Why is the temperature important when it is optimum temperature? Why is temperature important? We just answered a question. Why is the temperature important?
For maximum
Because
enzyme
it increases
activity.
the
Yes, of course, for maximum rate of reaction. All of them for maximum rate of reaction, actually. How do I control the temperature in any area?
Heater.
Yes, I will use a heater, an air conditioner. Okay, that's it. That's enough. Just one sentence about every point. Fertilizers that contain ions, they should contain what, for example?
Nitrate. Magnesium.
Very good. They should contain nitrates so that the plants will use them to make proteins. Okay, no pests. So I will not have any pests feeding on my plants. So I will have more crops or less crops.
More.
More crops. And I'm going to provide perfect carbon dioxide concentration. Again, because carbon dioxide is a limiting factor. If it is not present, it will affect photosynthesis. So I'm going to do everything to maximize photosynthesis and make the plant happy. Okay, glass houses, they have been already removed from the syllabus. But why am I answering this question? Because they can ask you this question as a suggest question. They can tell you information about the glass house. They will tell you it is a house where all the factors are provided. So what are the advantages or what are the disadvantages? What if they ask you about the disadvantages of glass house? What will you say? Give me one or two disadvantages of glass houses.
Diseases are easily spread.
You can say that if the glass house contains one type of plant. Also, what about the cost?
Expensive.
Of course, it is expensive. Okay. And how will this affect biodiversity? More or less? If everything is controlled and everything, and you are interfering with everything, and you are producing only one type of plant, this is going to increase or decrease biodiversity. What do you think? What, I lost connection again? Glass houses, they increase or decrease biodiversity. What do you think? And if I'm growing only one type of
Decrease.
plant, decrease, yes. I want you, while you are answering questions, think of what if they ask you the question in this manner or this way, so okay, to increase your chances of just like being exposed. Okay, here we have ecology. Outline the reasons why large mammals, such as some species of rhinoceros, are endangered. This is a very easy question. Why can the giraffe be endangered or the rhinoceros be endangered? Give me 5 reasons.
Habitat
What?
destruction,
Yes, I can
overhunting,
destroy their homes. Very good. This is a very easy question. I can destroy their homes. What else?
So the
overhunting,
screen isn't sharing.
Oh, it's not shared? Seriously? Yes, overhunting. Very good. 1 minute. Okay. Of course, overhunting. Overhunting. Okay. I will destroy their homes. I will destroy their land. And this is deforestation. It's very important. What else? What can cause any mammal to become denatured? Hmm? Where did you go? Hello? Hello? Are you with me? Can you hear me? Can you see my screen?
yes,
We're, we're nearly finished. Yes. Why can any animal become denatured? Very easy question.
no enough resources for food,
Yes, no food, no water. There's a natural disaster. There is war. I broke
floods,
down their, yes, there are floods or drought, the opposite of flood. Okay, we destroyed their homes. We broke down, we did deforestation. Why did we do deforestation? Why did I cut down the trees and huh?
to have places to build new buildings.
Exactly. When every time you mention deforestation, mention why you did it. Deforestation, I mean, I'm going to cut down trees and trees and trees. Why? Because I want space to build homes, to build roads, to build hospitals, whatever. Okay. People are not educated, no food, no water. Any one of those will be the answer. Discuss the problems that using such closely related animals might have for conservation of rhinos. Now, this is really important. And this is a common question. When I have rhinoceros and they are endangered, and then I'm going to keep them together. Let's say I have 4 brothers and 4 sisters of rhinoceros only, and I will keep them together. I am keeping them together to conserve them. But there are some problems when I use closely related animals in the conservation of any animal, in their conservation. What is the problem here? Oh, this is not what I want.
No variation. Diseases can easily
Yes,
pass.
yes. Look, I want to find it here for you. There's a list here that I want to highlight for you, and I want you to study it. 1 minute. Where is it? Where is it? Okay. Reasons for conservation programs, artificial. Here. Okay. This is in the lesson, Human Influence on the Ecosystem. I don't know which page because I am making changes and I removed the page numbers now. Endangered species. Please memorize those 9 points. Explain the risks to a species if its population decrease in size. This is a clear objective in your syllabus. An endangered species is at a risk of becoming extinct due to overhunting, predation, climate change, everything we said, loss of habitat due to deforestation. So this is the question that we answered. This objective is the answer to this question. Where is the question? Where did it go? This is the answer to this question. Question A, the reasons why large mammals can become endangered. So this is those 9 points are the answer to this question. Now, the remaining objective, endangered species can be helped by conservation methods. No, here. Risks to a particular species as a result of a decrease of its population size. I have very little number of rhinoceros, very little number. What is the risk? What is the problem when I have a very little number? This or those 6 points. I have a very small population, so they will breed together. Brothers and sisters will breed together. This is called inbreeding. And this is going to reduce biodiversity. Okay? This is breeding between close relatives. And I think I gave you the example when you have dogs and you have a litter of puppies, and those puppies, they breed together. And this is not something, this is not an advantage because this will reduce biodiversity. This would reduce genetic variation. This would increase the chance for genetic diseases. Any reason that will reduce biodiversity is always a disadvantage. They're not adapted to survive if there are environmental changes. If there is a disease, they will all be affected. It's just like the same disadvantages for asexual reproduction, because inbreeding, when you have relatives that are breeding together, they will be the same, very, very close. Okay. And when they're very, very close, this is going to reduce biodiversity with all the disadvantages of a decreased biodiversity. Let's read it together. Increase the risk of genetic diseases or mutation, reduced ability to survive to changes in the environment, higher chance of extinction. If there is a disease, it will spread. They are very small population, difficult to locate each other. They are isolated. They cannot find a mate. They're all too old to breed. Okay. So okay, I lost connection for a minute, but did you listen to this question?
Yes.
Important, is common. What is the exactly the same answer? Exactly the same question. What are the disadvantages of self-pollination? What are the disadvantages of asexual reproduction? Self-pollination is an example of sexual reproduction, but the plants, the flowers on the same plant are pollinating themselves. There is no external gene. There is no pollination from another flower. So there is a decrease in variation. Here I have a few number of rhinoceros. Okay, a few number. Let's say there are brothers and sisters, because animals, brothers and sisters, they can breed together. This is not good. This is called inbreeding. And the disadvantages are the same. Are you with me? Is the screen presented? Okay, so please understand this question. The same disadvantages of self-pollination or asexual reproduction. Why? Because here we have a case of decrease in the biodiversity. I have a few number of rhinoceros. They are brothers and sisters, and I want them to breed together. In breeding in animals means I'm going to breed brothers and sisters. And this would decrease variation. Decreased variation has a list of disadvantages. A decrease in the genetic pool, an increase in the chance of genetic diseases being transferred. All the alleles will be the same, they will all be homozygous. There is an increase in the chance of extinction. There is spread of disease. They will be infertile. They're too old to breed. Please memorize the list. Okay? I hope I'm clear. I said it 3 times. Outline the process of artificial insemination. Who will remind me? What is artificial insemination? This is one way where they can conserve endangered species. We do
When the sperm is collected from the male donor and is washed and screened
very good.
and is inserted through the female cervix during ovulation period.
Very good. Excellent. During her fertility period. Here it is different from in vitro fertilization. In vitro fertilization, we take the sperm and the egg out. But in artificial insemination, the egg stays inside the female. I only take the sperm, I will screen it, wash it, and I will insert it inside her body at the right time when she's ovulating so that I will increase the chances of fertility. Excellent. And we do this when we have couples that cannot have babies or with endangered species so that they don't get endangered. Okay. What is the part of the human gas exchange system that will contract to decrease the air pressure in the lungs? Who
Diaphragm.
remembers? Very good, the diaphragm. And the diaphragm, we said they are friends with which other muscle?
External intercostal.
Yes, external intercostal muscle. I hope you don't forget that. The air flows between the larynx and the lungs. What will take the air from the larynx to the lungs? It's so easy.
Trachea.
Okay, okay. Now, what will contract to lower the ribs in forced expiration? Out. What will contract when you are exhaling?
Internal.
Yes, internal intercostal muscles. Now, if you did not study this lesson and you forget this part, you are in trouble. What is the site of gas exchange?
Alveoli.
Very good.
Alveoli.
Please all participate, not just Selma. Now here you have another graph for the 10th time. You have 2 graphs on one grid. Okay, of course, look at the key here is the rate of breathing and the rate and the depth of breathing. Describe the effects of changing the carbon dioxide concentration of the air on the rate and the depth of breathing as shown. Now, this is a question that has got nothing to do with how much you studied because they're talking about describe only, no explain. So you will go to the graph and you will describe every single part of the graph with numbers, with numbers, with numbers. You have to use numbers. They did not give you the graph to waste time. So you will go to the graph. You will see that you have 2 grids, one for the rate of breathing and one for the depth of breathing, and see how they will change with time. Both of them at the beginning, they were, what's the percentage that change?
0.
Percentage change was 0. This does not mean that the person doesn't breathe, because also someone sent me this question and told me this means that the person is not breathing. No, habibti, they're breathing. But the percentage change, there's no change. They're breathing, but there is no increase or decrease. And then at a certain time, you will write the time. This is nearly 5 minutes or close to 5. What happens to both of them?
Increase.
They both increase. And what will increase more than the other? Look, it's obvious that one is more than the other. Which one? The
Breathing
rate or
rate.
the depth? The rate of breathing. Okay, the solid line. So both of them will increase, but one of them will increase faster than the other. And give numbers, give numbers, give numbers. And then they will reach a peak. Okay? They will reach a peak here, and one of them will reach a peak here and will become constant for a short time. Again, give numbers. Okay, the percentage change, the peak. Give numbers, give numbers, give numbers. And then both of them will hmm,
Decrease.
both of them will decrease. And again, one of them will decrease more than the other. Okay, so you will take the graph and you will break it down bit by bit. The depth of breathing will decrease more, and you give numbers also for the 10th time. And then they will both increase, or the depth of breathing will increase and go back. And the rate of breathing will become constant. So this is so easy. Any graph, you have lines either going or constant or going down. They just want to know if you know how to read a graph and give numbers and compare. If you have two graphs, one is above the other, so A is growing. They're both going up, they're both increasing, but A is faster than B. If you have two graphs going down, then B is going down or decreasing faster than A. If you have two graphs that meet at a certain point, then they both meet at for a certain temperature or a certain point or a certain whatever. Okay, they meet. This is when two graphs meet. When I have maximum height, this is optimum. Okay, so I think you know how to describe graphs very well, and numbers, numbers, numbers. Now let's finish this exam that doesn't want to finish. Complete the sentence. During physical activity, the carbon dioxide concentration in the blood increases. And the reason for this is an increase in the rate of what? What increases CO2 in the blood? Where did you go? You're there, and I did not lose connection. So, what increases CO2 in the blood, Yusuf?
Respiration.
Yes, give me an answer. I don't know why you're too lazy to speak up. Okay, respiration. Here, where is the exam? It went. Yes, the respiration that occurs in muscle cells. Aerobic respiration. What takes place? Which organelle inside the cells that do respiration? Huh?
Mitochondria.
Excellent, mitochondria. Okay. The increase in concentration of carbon dioxide in the blood is detected by the, who will detect it?
Brain.
The brain. Okay, good, easy. This is easy. Which increases the rate and depth of breathing so that carbon dioxide can be, keep it or,
Excreted,
yes,
expelled,
I want to remove it. Yes, very good. This is important because carbon dioxide is a what substance that can cause harm?
toxic.
Toxic. It reduces pH of the blood. We took it, blah, blah, blah. Okay, this is easy. Now, finally, the last question. Now we have a Punnett square. We have the parental blood groups A and B, parental genotype. This is heterozygous, and this is also heterozygous. And okay, what will you do? How will you distribute the genes here? Tell me, I forgot.
Every
This is the gene.
gamete square.
Yes. Very good. This is an gene made up of two alleles. I'm going to break this down. This is the O, and this is, or you can just, if you want to, you can do it like this. It's still correct, but I prefer that you do it exactly like it is given to you. Just A and O or B and O, but let's use the same format. Okay. Now I'll do this quickly. This is going to be AB. I'm sure you know it. If you have any issues, ask me. And this child will have blood type B. This will have A. Always write the dominant allele first. And here, homozygous recessive or blood group O. What are the phenotypes of the children? Very easy. I'll write the phenotypes
AB.
of the children. Yes, AB, comma, B. This is the genotype. So you just write B. They want the phenotype. A and O. Okay? What is the probability that the child will have blood group O? What's the probability or the percentage or the ratio?
25.
25.
Yes, you're all with me. Okay, very good. Or 1 out of 4. Explain why ABO blood group system is an example of codominance. Nice question. Also direct question, thankfully.
Alleles are all expressed in the phenotype.
All alleles,
Most, yeah.
if you have the two dominant alleles, A and B are dominant. Okay? When both dominant alleles are present in the genotype, the phenotype will be AB. So when do you have a co-dominant condition? When you have two dominant alleles and they are both expressed in the phenotype. Okay? When these two alleles, both of them are dominant, and both are expressed in the phenotype. This is an example of codominance. But what you said is right also, okay? Because the O, if it is present with a dominant allele, it will not be expressed. If you have AO, the blood group will be A. If you have BO, the blood group will be B. So when will all the alleles be expressed? When they are dominant, when they are all dominant, like A, B. A is dominant, B is dominant, and they are both expressed. Okay, this question doesn't finish. With reference to the figure, explain why ABO blood group system is an example of discontinuous variation. Very nice question. You forgot discontinuous variation? You will have a problem. What are the features of a discontinuous variation? Is the blood group affected by the environment?
No.
No.
No, not affected by the environment. This is so important. Not affected by the environment. It is affected only by the,
By
Genes.
the genes.
very good, only by the genes. Now, what is the definition of discontinuous variation? What do you see from the graph? You have a range of phenotypes or limited phenotypes?
A limited
Limited.
phenotypes.
Yes, limited phenotypes with no categories in the middle, with no examples, with no intermediaries, with no, like I have either A or B, I have nothing in between, okay? With no examples or no categories between A and B. So you will write all the features of discontinuous variation. Affected only by the gene, not affected by the environment. I have limited number of phenotypes, A, B, AB, and O. Only four. I have only four. I don't have any more phenotypes. And I don't have anything in between. No categories in between the blood types. Okay, so this is the only direct question that I saw on this exam. Okay, the exam is finished. This exam is a little bit tricky. Okay. What was tricky? The inheritance question was easy. The graph question was easy. This question here, ecology questions were not easy. Please study ecology. Please do not ignore ecology. Ecology is found in every single exam, every single variant, and at least 2 essay questions. So at least it's out of 8 marks or 6 marks, 3, 3, 4, 4. Ecology is very, very, very important. Maybe it's the most important lesson. And it's easy because you only memorize it. Okay? Any questions over this exam?
No.
Fine. Any questions in general? Do you want me to revise anything?
No, thank you.
Okay, I am still with you anytime. If you're solving an exam and you find a question that is tricky, send it to me like you do. I'll answer you if you send me anytime. Okay, best of luck. And if I meet you again soon, I'll tell you before the exam. Okay? Bye-bye.
Okay.